Okay, so the excitation contraction coupling process that we see with smooth muscle. And again, I really want you to be pointing out some of the differences or understanding some of the differences that I'll point out. So first of all, we know we have a smooth muscle cell here. We do not have that traditional neuron-to-neuron synapse. We have varicosities that are interacting with this smooth muscle cell, the spindle-shaped smooth muscle cell. Now, the first part of this process is understanding that most of the calcium that drives smooth muscle contraction comes from the outside of the cell. All right, so we got lots of calcium outside the cell. And we did not see this with skeletal muscle, right? Most of the calcium from skeletal muscle came from where? The sarcoplasmic reticulum, right? The SR. Most of the calcium here comes from outside of the cell, okay? On the membrane of the cell, we have voltage-gated calcium channels. And so when the membrane potential of this cell changes, that triggers the opening of these calcium channels, and this calcium can rush into the smooth muscle cell. Okay, so that's sort of the second process here, is that the membrane gets depolarized. The membrane gets depolarized, and we have that rush of calcium coming in because this voltage-gated, let's write it here, the voltage-gated calcium channel opens up, okay? All right, now, inside the cell, we also have some calcium coming from the sarcoplasmic reticulum. Okay, and so the third part of the process is when the intracellular calcium begins to rise, we have the opening up of voltage-gated calcium channels that are on the membrane of the SR. So there are really two sources of calcium that are really important for smooth muscle contraction. Most of it coming from the ECF, right, outside of the cell. But then the third step here is that we have calcium-induced calcium release, meaning that that initial flux of calcium that came in from outside the cell triggers the opening up of these voltage-gated calcium channels on the SR membrane, and that allows more calcium to get into the cytosol, okay? So the third step here is calcium-induced calcium release. And that really floods the cytosol with all of this calcium, all right? Now, the fourth step is that the calcium must bind to this protein that's called calmodulin. So calmodulin is in the cell. Now, in the absence of calcium, calmodulin is doing nothing. It's turned off. And so the binding of calcium by all of this calcium that's now flooding the cytosol is what kind of turns calmodulin on, right? This is a calcium, binds to calmodulin, and this creates a calcium calmodulin complex. Okay, so all this calcium rushes in, it's able to access these calmodulin proteins, bind onto it, activate them or turn them on, and they're gonna go and do something else, but understand that in the absence of calcium, calmodulin is dead, doing nothing, okay? The fifth step here is that this calcium calmodulin complex is going to activate another enzyme in this cell, right? This enzyme is called MLCK. MLCK, and that stands for myosin light chain kinase. Okay, and so this complex is what activates this myosin light chain kinase, right? Now, what did we say a kinase does? What's the job of a kinase? What does a kinase do? Can we see this? What does a kinase do? It's going to phosphorylate, right? It's going to add a phosphate group. We talked about this covalent regulation many moons ago in Unit 1, where we can turn something on by phosphorylating it, we can turn it off by dephosphorylating it, or vice versa. And that's what the job of this myosin light chain kinase is. When it's activated by this complex, it is going to go around and look for the myosin heads, and it's going to be able to turn them on, right? Phosphorylate them and turn them on. So step 6 here is that we phosphorylate the myosin heads, okay? And that leads us to cross-bridge cycling, which we know translates to muscle contraction, right? Muscle contraction. And so just to remind ourselves, we said that in smooth muscle, we don't have T-tubules, we don't have troponin, right? We do have actin and myosin, okay? We do have those things, and so we still see the sliding filament model, but it's brought about in a very different way, and it's regulated in a very different way than it was for skeletal muscle. So the job of calcium here is to bind to this complex, turn this MLCK on to then phosphorylate myosin heads. When the myosin heads are phosphorylated, they're in their energized state, and they can go and form those cross-bridge cycles, and that leads to muscle contraction. Now, in order to cease this muscle contraction, there is another enzyme that is lingering in this cell, and these are called phosphatases, okay, phosphatases. Can you see this? And so just like we said, a kinase is going to add a phosphate group, a phosphatase is going to cut or cleave that phosphate group off. Now, understanding how these two are regulated really relies on understanding the role of calcium here. So in the presence of calcium, we have this calcium calmodulin complex, which turns MLCK on, but when the calcium leaves the cytosol, right, gets pumped back into the SR and back out of the cell, right, that's another important component here. We got to pump it back out of the cell and back into the SR. Then we have the activation of the phosphatase. So basically, when we have decreased calcium, we turns on these phosphatases. And that is going to dephosphorylate the myosin heads, okay? Dephosphorylate the myosin heads. And that is going to lead to cessation of cross-bridge cycling. And this is going to lead to muscle relaxation. Okay? So let's go over this process one more time from the top. So the main source of calcium that drives this process is coming from the ECF, right? I'll write it out here. The ECF. And when the membrane of this cell gets depolarized, this triggers the opening of these voltage-gated calcium channels on the membrane of the cell. That allows an initial rush of calcium into the cell. Now, that initial rush of calcium further triggers calcium from the SR. So again, the calcium opens up this voltage-gated calcium channel on the SR, and so we really flood the cytosol with calcium. That calcium goes and binds to calmodulin, and that creates a calcium calmodulin complex, which can then go and activate MLCK, this enzyme, myosin light chain kinase. Now, when we activate or turn on MLCK, MLCK will then phosphorylate or add a phosphate group to the myosin head, and that will start or initiate cross-bridge cycling. That's how we get muscle contraction. Now, when this needs to stop, the calcium leaves the cytosol. It's going to be pumped back out of the cell into the ECF. It's actually going to be exchanged for sodium. I'll talk about that in a minute. It's going to be exchanged for sodium, and then it's going to be pumped back into the SR. And so there's no calcium in the cytosol. And the lack of calcium is what activates or turns on the phosphatase. And the phosphatase will go around and grab the phosphate off the head of those myosin heads, and that will cease cross-bridge cycling, hence ceasing muscle contraction, okay? So again, I'm going to point out, it's really important to understand what calcium is doing here. Calcium is also turning this on and turning it off, just like it did in skeletal muscle, but it's binding to something different, right? It's binding to calmodulin to bring that about. So when it binds to calmodulin, it turns this on. When it's out of the cell and it's not on calmodulin, it turns this off, okay? So it's still regulating muscle contraction here, but in a very different way.